So hello everyone. First of all, I'm sorry for my voice. Today we are going to talk about the nucleus, cell cycle regulation, and cell death. Why this topic is so relevant nowadays? Because different problems with the cell cycle regulation result in the formation of tumors. And to understand how to treat different types of malignancies, we have to understand how normal cell cycle is regulated, what to repair. After that, this topic is very relevant because we are approaching to the gene therapy and to the gene editing in humans. Of course, this is not the therapy of modern medicine, but you know the genetic modification is widely used in plants, in animals, and nowadays in humans, modern vaccine against the coronavirus offered by Pfizer, it contains the RNA, nucleic acid, and in my humble opinion, this vaccine it solves several issues, not only like prevention of coronavirus infection, but also it helps to organize all the infrastructure to distribute DNA in purposes of treatment. So I think that in this way mankind is preparing for the gene therapy in the nearest future. In any way, you have to be aware about the normal regulation of events in the nucleus, and let's start to investigate it. So first we are going to talk about the nuclear envelope. So nucleus is a very important a center of the cell, as it contains the information about the structure of the whole organism, not only of this particular cell, but about the whole organism. And it should be properly insulated from the cytoplasm. And to do that, it has two membranes. The outer membrane is continuous with a rough endoplasmic reticulum, so different ribosomes could be found on it. And the inner membrane is faced to the nuclear plasm itself. Both membranes, they fuse in the region of nuclear pores. And nuclear pores, these are like gates, and they allow the exchange of substances between the cytoplasm and the nuclear plasm. So the space between the two membranes is called the perinuclear cistern, and it is continuous with the cisterns of the rough endoplasmic reticulum. Then do not forget to say, when you are talking about the nuclear envelope, about the nuclear lamina. This is the network of intermediate filament called lamin under the inner membrane. Exactly, nuclear lamina helps to maintain the shape of the nucleus and helps to fix chromosomes inside. And to understand better the function of the nuclear lamina, let's look what happens if those proteins are mutated. There is a very rare disease, which is called progeria or premature aging, which results in a premature aging of children. Why? Because they have defects in the nuclear lamina. This is how their nuclei look like under the fluorescent microscope, comparing with the normal one. As you can see, normally nuclear lamina is rounded and maintains the stable shape of the nucleus. When those proteins are mutated, the nucleus is of regular shape, and such cells they can't divide effectively. So the process of mitosis is violated, and as a result, the regenerative potential is very restricted. So such people become aged very early, at age of 10 years. Okay, so let's move on. After we have discussed two membranes and the nuclear lamina, let's talk about the nuclear pores. These are very important complexes, as they not only allow exchange of substances, but they control and regulate what substances to allow to enter the nucleus, what substances are not allowed. So first, how this complex is organized. It has the central framework made up of 8 granules. Ring made up of 8 granules. It is inserted between the two membranes, and also 2 additional rings: cytoplasmic ring, also made up of 8 subunits, and nucleoplasmic ring, also 8 subunits. And from the cytoplasm, we can see that protein fibrils extend to the cytoplasm and to the nucleoplasm spikes extend, forming the so-called basket. Like in basketball, here we can see the same structure. So if we are talking about the transport, we have to mention two types of proteins assisting in this transport: importins and exportins. As it is obvious from their name, importins, they help to involve something inside the nucleus, while exportins, they help to export. And very important element of this process is that how importins distinguish what proteins are allowed to enter the nucleus and what proteins are not. It depends on the protein itself. If protein is destined to perform some important function in the nucleus, such proteins, they have a special amino acid sequence, which is called nuclear localization sequence. As you remember last lecture, we were talking about the ribosomes sitting on the rough endoplasmic reticulum. There were a signal recognition particle. It recognized the special amino acid sequence. If protein is destined to endoplasmic, Golgi, or lysosomes, they have a special sequence recognized by that particle. In the same way, if protein destined to be located in the nucleus, such proteins are produced in the free ribosomes, not associated with the rough endoplasmic, and such proteins they have a special amino acid sequence called nuclear localization sequence. It is recognized by importins, and then with the help of GTP energy, substances are transported to the nucleus. And to leave the nucleus in the same way, substances should expose nuclear export sequence. So if there is no such sequence, then particle become entrapped in the nucleus. So to leave the nucleus also, they should have this pass. Okay, let's move on. And now, as we have discussed the border and how the permeability is regulated in the nucleus, we should talk about its content. So what's there inside the nucleus, why it is so important, and why this is the most important organelle in the cell. First, we have to imagine several numbers. The human genome was deciphered, and it was revealed that we have a little bit more than 20,000 genes in our genome. Protein coding genes, a little bit more than 20,000. If we compare our genome with the genome of this small worm has even more protein coding genes. If we compare the number of non-coding genes not coding proteins, we'll see this huge difference. And we have many more non-coding genes comparing with this worm. So it means that DNA, it has not only protein coding function, but some other. And we also can understand that the evolution was driven not towards the increased number of genes or proteins or their diversity. We did not diversify our protein spectrum, but instead we've complicated the system of mastering those proteins. So the system regulating the activity of those proteins was evolved and developed along the evolution. So another several numbers. The total length of DNA packed in the nucleus of only one cell is nearly 1.5 meters. So this rather long DNA molecule is packed in a very small nucleus of only several micrometers in diameter. So it should be very tightly packed. At the same time, this information should be used, not all the information of course, but every cell uses some special genes, But if we analyze what is the percentage of protein coding regions are there among those 1.5 meters, we'll reveal that less than 2% of its length encodes proteins, but more than 90% of human DNA is transcribed into RNA molecules. So what I want you to remember, to understand, the DNA has the function not only to encode proteins but also to produce RNA molecules, and there are many more than just transport, messenger, and ribosomal RNAs. There are small interferential RNAs, nucleolar RNAs, and many more, and they perform regulatory function, and this regulatory function even predominate over the number of proteins. DNA, it performs very important function, and there are a lot of genes, a lot of functions, and they should be properly organized in a very small nucleus. So how it is possible? It is possible because every cell, it uses only small amount of the information. Because, for example, cell in the brain, neuron, for example, shouldn't know the structure of the myofibril, which is normal for the skeletal muscle cell, and vice versa. So every cell uses only some part of genetical information. All other information is tightly packed and shifted to the periphery of the nucleus. So to understand how our nucleus, DNA is organized in the nucleus, we should understand the hierarchy of its package. So the first level of DNA package, this is the double helix, this is the DNA itself. After that, it is coiled and wrapped around the histone proteins. So nucleosome is the next level of organization. Here it is shown. What is it? Every nucleosome is represented by 4 pairs of histone proteins. In general, there are 8 of them: H2A, H2B, H3, and H4. A pair of each forms the core, around which 2.5 turns of DNA molecule could be found. So together they are called nucleosome, and H1 histone protein is used to attach them. So by wrapping DNA around histone proteins, we decrease its length. After that, 30 nanometer chromatin fibril is formed by coiling those nucleosomes around its axis. So if we compare the nucleosome with a necklace, then this necklace beads on a string, they turn into spiral manner, and this spiral structure is called 30 nanometer chromatin fibril. By coiling them, we decrease their length, so we become even shorter and more tightly packed. After that, this 30 nanometer fiber get attached to the scaffold protein. As you can see, this green color scaffold protein, it is shown. This is the part of the nuclear matrix. So inside the nucleus we have 3-dimensional network of scaffold proteins. They are like ropes extended and oriented between the 2 opposite nuclear pores, and chromatin 30 nanometer chromatin fibrils are attached to those scaffold proteins in the nucleus, and usually attached regions are actively expressed, actively used by the cell, while those loops are not used, and they form the heterochromatin. We are going to discuss further. So loop domains, those loops of 30 nanometer chromatin fibril, this is the next level of DNA organization, and by forming those loops, DNA is even more coiled, as you can see here. They are shown, and in this way the total length of DNA is decreased. So if we look under the microscope at this DNA molecule, we would not see their DNA, it is very small, but instead we'll find here light and dark regions in the nucleus. Both are occupied by the chromatin. So chromatin, this is the complex of DNA, protein, and RNA. But in some regions they are transparent, the chromatin molecules are transparent, and in some regions it is dense. So we distinguish euchromatin and heterochromatin respectively. Euchromatin, this is a loose, light, electron-lucent, and it is actively used by the cell. As here, DNA is uncoiled and it is exposed to the action of different enzyme and transcription factors. While heterochromatin is tightly packed and it is dense, coiled, and it is not used by the cell. There are two types of heterochromatin: constitutive, which is not used at all, and facultative, which could be used under some circumstances. So some of those heterochromatin regions could turn into euchromatin if special circumstances would stimulate it to do that. So also depending on the location of this heterochromatin, we distinguish them marginal, close to the nuclear envelope, then perinucleolar, here around the nucleolus, and some karyosomes. These are the patches of heterochromatin among the euchromatin. Yeah, karyosomes. Karyon, this is the nucleus in Greek. So karyosomes, like nuclear bodies, is translated. Okay, so this is how we can see the chromatin. So chromosomes are not visible when cell is performing its function between the divisions. And notice that in the region of nuclear pores, heterochromatin is absent. So when something is entering, heterochromatin should not block the nuclear pore. Okay, and an example of the heterochromatin is a Barr body. Barr body is facultative heterochromatin present in females, and it is represented by the inactivated X chromosome. So the X chromosome is inactivated randomly in different cells of our body in the embryonic period. So in blood smear analysis, this chromosome forms a Barr body, a special segment in the neutrophils, and by the blood smear we can detect and distinguish females from males. When we have the topic blood, after several lectures we'll return to that. And today I want to ask you a question for the most curious students. Here in this picture, two cats, female cats, are shown. This is the prototype and he is the clone. And the question is, why two genetically identical cats, and they are really genetically identical female cats, why this one has another distribution of patches comparing with this? Why genetically identical female cats, they have different distribution of colors of their fur? Okay, and if you know the answer, please write down in the comments, and next lecture I'll tell you, at the beginning I'll tell you the real answer. Okay, so let's move on, and we have to discuss today a lot of things. So nucleus, here the brief video, the full link is here, and it shows us nucleosomes with those purple color. Notice that histone proteins, they have tails extending out of the nucleosome, and notice that actively transcribing regions, and here we can see RNA polymerase produces RNA, they are uncoiled. And here nuclear pore is shown, and you can see this basketball structure of the nuclear pore face to the nucleoplasm. This is the cytoplasmic part of the same pore, and it differs as it has fibrils on the surface. Okay, next, what we have to say when we have discussed the chromatin is that chromosomes are distributed not randomly in the nucleus, but each chromosome, it has its own territory. And to exploit and to use the information which is there in the chromosome, it is very important to put it properly in the nucleus. And here you can see the fluorescent in situ hybridization method, when each chromosome is shown with its own color forming a map inside the nucleus, and here the distribution of different chromosomes are shown. Notice that even the chromosome of the same pair, for example, the 9 chromosome is here and there, we have a pair of each chromosome, paternal and maternal. So even chromosomes of the same pair, they have different position. And it is very important because if we change the position of the chromosome, it will be used improperly. And despite we have a normal DNA sequence in the gene, if this gene is put in another place in the nucleus, it will not be used. So the information encoded in the nucleus should be on its own place, like in the libraries. If you come to the library and ask librarian to bring you a textbook, this textbook should be on a proper shelf. If we change the position of the textbook, then librarian would never find it. In the same way, our nucleus is organized. So, also another term you should know, this is the epigenetics. When we're talking about the nucleus, you should know that histone proteins, they perform their function not only in organization of DNA, so they perform not only structural function by coiling the DNA, but also they extend their tails outside, and those tails of histone proteins could be modified by acetylation or methylation. Many other modifications are possible also, but those two are the most widespread. And by either acetylating or methylating those tails, the chromatin could turn into heterochromatin or euchromatin. And in this way, by modifying those proteins, we can stimulate genes to be transcribed, or instead we can repress those genes. So epigenetics, this is the combination of different types of histone modification or DNA modification, regulating the activity of genetical information. So sometimes, why it is important, because sometimes even if the nucleotide sequence is normal in the gene, if it is improperly modified, or if locally histone proteins are improperly modified, then such genes could be repressed, put in a heterochromatin, and never used by the cell. So the problem is not with the nucleotides, and the problem with epigenetics. And different syndromes nowadays, they are very well known, develop not due to problems with the sequence of DNA, but due to the epigenetical modification. That's why you have to know about that. Okay, and then nucleolus. Nucleolus is the region of the nucleus. So we summarize the information about the structure of the nucleus. And nucleolus, this is the most dense region of the nucleus, and it is not bound with a membrane, and it is the place of ribosomal RNA synthesis. In general, nucleolus, it is translated like a small nucleus. Your nucleolus is organized by 5 chromosomes, 13, 14, 15, 21st, and 22nd. So all those chromosomes, they have regions with genes encoding ribosomal RNA, and they come close one with another and form so-called transcription factory in the nucleus. What does it mean, transcription factory? This is the region of the nucleus where functionally related genes come close one with another, and specially they are organized into this transcription factory. Okay, so inside the nucleolus, we can distinguish fibrillar centers. These are those regions of DNA. Then fibrillar material. These are the actively transcribed ribosomal genes with the formation of ribosomal RNA. And granular material. These are the newly formed ribosomal subunits, some small dots. And exactly because of the granular material, nucleolus is so dense. So nucleolus is not a part of heterochromatin, okay? Despite this is the dense region of the nucleus, this is not a heterochromatin, this is euchromatin. But its density is due to the presence of many ribosomal subunits in it. Okay, also one more term you should know. This is the nucleolenema. Nema means a thread, and nucleolenema, this is the network formed by fibrillar and granular material taken together. It's very easy to see. You can see that nucleolus under high magnification is made up of something like lines. And these are exactly nucleolenema. Yeah, and one more protein you should know. This is the nucleostamine. Stamine because it's characteristic for stem cells. This protein, it binds p53, and it is characteristic for the regulates the cell differentiation. So more differentiated cell is less nucleostamine it has. So nucleostamine is more characteristic for stem cells, undifferentiated cells, and as it is associated with the nucleolus, it is mentioned in this slide. Okay, now let's talk about the chromosomes. So we have just discussed the chromatin, what regions of chromatin, what kinds of chromatin could be distinguished, how DNA is packed there, and now let's discuss how chromatin looks like during the mitosis. It forms chromosomes, and chromosomes, this is tightly packed chromatin, destined to divide the genetical information between the two daughter cells. So in every chromosome, during the division, we can distinguish the centromeric region and the telomeric regions. Centromere is located in the center, it's obvious from its name, and centromere doesn't encode proteins or ribosomal or any RNA molecules. In the centromere, we can see many repetitive sequences, and they are not conservative, so they are not very important. Centromere performs structural mechanical function, as this is the place where two sister chromatids attach one with another, and also this is the place where mitotic spindle fibers attach to chromatids. Okay, at the end of the chromosomes, we can find the telomeric regions. Telomeric regions, they also do not encode proteins. They also made up of repetitive sequences of DNA, and they become shorter with each next cell division. So telomeres are needed to count the number of divisions, because this number is limited. We'll talk further. So by the position of the centromere, we can distinguish different types of chromosomes. That's what you have to know. Metacentric means that centromere is in the center and both arms are equal in their lengths. Then submetacentric, when centromere is shifted to one of those two poles. Then acrocentric, when centromere is when one arm is much shorter than another, and telocentric, not in humans, when one arm is completely absent. So why it is important to distinguish those chromosomes? Because there is a special diagnostic method which is called karyotyping. In this method, all the chromosomes are counted, and they are assessed. Their quality is assessed. Karyotyping helps to reveal different types of chromosomal aberrations. If there is a spare chromosome, or one chromosome is absent, or a part of one chromosome is translocated to another. To understand that, how this procedure is performed. So once again, we have different types of chromosomes. We have 23 pairs of chromosomes and 22 pairs, and also sex chromosomes. If the number is not normal, it results in different genetical defects. So to reveal those defects, karyotyping is performed. How it is realized, this method? A droplet of blood is taken from the patient. Then this blood is incubated for 2 or 3 days with a special substances stimulating mitotic division in leukocytes. At the same time, special chemicals like colchicine are added to break down the mitotic spindle. So cell is stimulated to enter mitosis, but at the same time spindle is broken down, so chromosomes are not split and they are not separated into two daughter cells. After that, this sample is centrifugated, cells are taken and other debris is removed. Then cells are fixed to stop the postmortal changes and put on a slide. At the same time, they are stained with a special fluorescent dyes to color different regions of chromosomes in different color. It helps us to distinguish them. Imagine those chromosomes are black and white. It will be much more difficult to organize them into pairs. And after that, they are investigated under the microscope. Karyotyping method helps us to understand is there any genetical pathology. Especially it is helpful in embryology, in the in vitro fertilization method, if we want to avoid chromosomal abnormalities, for example if parents are aged already, and that's what we are going to discuss next lecture. Okay, let's move on. After we have discussed the centromeric regions and types of chromosomes according to the position of the centromere, we have to discuss telomeres. This is extremely interesting region. Telomeres repetitive nucleotide sequences. They do not encode proteins, but they become shorter and shorter with each next division. And under normal circumstances, every cell of our organism has a limit, which is called Half-life limit, is nearly 50 divisions. So every cell can undergo not more than 50 to 70 divisions. Okay, after that, telomeres become extremely short, and such cell can't divide anymore, because otherwise it would lost a significant DNA sequences encoding proteins, for example. But if we are talking about malignant cells, they become crazy and they do not stop, they divide all their life. So this mechanism of limiting the cell division is very important to prevent different mutations in our organism. And another function of telomeres, except counting the number of divisions, is to fix chromosomes to the nuclear lamina. We have already discussed that every chromosome, it has its own territory, and every chromosome is fixed to the nuclear lamina. To fix chromosomes, exactly telomeres are used. So what is especially interesting is that some cells, like our gamete cells, stem cells, and tumor cells, they have a special enzyme called telomerase. Telomerase it regenerates telomeres after the division. So cells with active telomerase, they have unlimited proliferative potential. That's why cancer cells, they do not die by dividing eternally. You know that cancer cells, besides many scientists, they think how to achieve the eternal life, how to solve the problem of death. And cancer cells exactly solved this problem, because they can live forever, but they destroy the organism in which they appear. That's because of the telomerase enzyme. Besides, what is especially interesting is that why telomeres restrict the number of cell division. Why cells are not allowed to perform more than 50 divisions? That's because with every division, cells, they replicate their DNA, and the replication process is not perfect, and there are some mistakes are made every time. So after 50th division, it is thought that the number of mutation is very high now, and it is better not to divide further, because further accumulation of mutations would result in problems. And that's why organisms, they become aged and they die eventually, because of those mistakes in the genome, like that. Okay, and what is interesting is that the telomere diagnostic nowadays is possible, and just it is interesting. I don't think it is really useful, but it is offered to estimate the length of telomeric region, and in this way to understand and to reveal the real biological age of the individual. Okay, and different even drugs are offered to stimulate, to boost the telomere length. I don't think it works really, but yeah, it's a commercial product. Okay, so to summarize, so the centromere is the just region in the center of, not usually in the center, but it's the region where two sister chromatids are attached one with another, and where spindle fibers are attached during mitosis, and kinetochore. Another structure you should understand, kinetochore is a protein complex attached to the centromeric regions during the division, and exactly kinetochore is used to organize microtubules. We'll discuss it further. Okay, and after we have discussed the nucleus with its chromosomes and chromatin, we have to discuss the cell cycle events, how nucleus is dividing, and how cell decides when to divide. First of all, we have to say that cells, they divide from time to time in adult organism as well as in embryonic period. So division is needed to regenerate some populations of cells, and some cells in our organism are not, they do not divide, for example, like neurons, and they belong to the static cell population, while other cells are rapidly dividing, for example, cells in our stomach mucosa, or cells of our epidermis, or cells in our red bone marrow. It depends on different factors, but what is interesting that the number of and intensity of cell renewal in the population correlates with the probability of tumors. For example, tumors very rarely arise from neurons, but very often they arise from gastric mucosa, for example. So higher the number of stem cells is in the tissue, the more high the probability of cancer in this tissue. And this is what is written in this article. I offer you to read because it's really interesting. So I want to highlight just why I offered you to read this article, because really every tumor is something like a crazy stem cell population. So we need to understand how normal stem cells regulate their activity and how they limit their division potential to understand how to stop tumor growth. Okay, so normal cell cycle, normal activity of cells regulated, and cell cycle, it includes the interphase G1 as G2 periods and mitosis. Cells, renewing cells, from time to time they re-enter mitosis to increase their population, not to increase, to maintain on a stable number. But some cells, they exit the cell cycle and they enter the so-called G0 period. So cells actively performing their function, actively working cells, they are in the G0 period. They do not divide, they are not preparing for the division, they just perform their function. For example, pancreas produces its hormones. So cells do not divide. But if it is needed, if the organ is damaged and regeneration is activated, then such cells, they can re-enter the cell cycle. So in cell cycle we have these phases, but some cells are in G0 phase. Okay. So, what about the interphase, about mitosis, you know a lot of from biology, but what about the interphase? Interphase, it includes the G1 period, before the mitosis, before the synthetic. In G1, cell, it store nutrients and is preparing for the DNA replication. For example, histone proteins are produced here. Then synthetic period, it includes the synthesis of DNA and duplication of centrioles. After that, G2 period, it includes the storage of tubulin molecules, and here cell prepares for mitosis. And after that, mitosis starts when cell divides into two daughter cells, and then daughter cells, they start their own cell cycle from the G1 period, or one of them or both could enter the G0 period and perform their function. Okay, but what is especially important is how cell cycle is regulated, how cell understands in which phase it should be right now. Cell cycle regulation is performed with the help of combination of two very important proteins, cyclins and cyclin-dependent kinases. That's what you have to know, and why those molecules are so important, because they are targets for modern chemotherapeutic drugs. So nowadays cancers and different malignant tumors are treated with the help of different molecules affecting cell cycle regulators. And in every phase there is a characteral combination of the cyclin and cyclin-dependent kinase. For example, for G1 period this combination is characteral, and so on. And the entrance of one phase and exit from the previous is regulated by changing of those combinations. First, those factors were discovered and called maturation promoting factor. Maturation promoting factor is the combination of the cyclin B and cyclin-dependent kinase 1 together taken. After that, many other cyclin and combination were discovered, and they regulate every phase. Okay, but what's especially important is how cell understands whether it can enter the next phase, whether it may not. There are different checkpoints, and that's what you have to know about. So in the cell cycle there is a spectrum of checkpoints, and every checkpoint is very important. It helps cell to stop and think whether everything's okay and whether cell is allowed to enter the next phase. The first checkpoint is the point of no return. This is the checkpoint of the G1 period. And before entering the synthetic period, cell should assess its size, amount of nutrients, whether it will be enough to replicate the DNA, because if the amount of nutrients is not enough, then there is no reason even to start the synthetic period. Okay, and the combination of retinoblastoma protein and transcription factor E2F, the combination of them both, they block the cell cycle propagation in case the cell is not ready. So those two proteins are very important. Then cell always checks the quality of DNA. If in G1 period, before synthetic period, different DNA damages are found, cell doesn't enter the S period. So before duplicating the DNA, its quality should be assessed. Then the DNA quality is assessed in the synthetic period itself and in the G2 phase. So it is very, very important before increasing the number of cells, the maternal cell 3 times checks the quality of its DNA molecule, because DNA is the most important thing in the cell. Okay, and if mistake is found here, there, or there, cell stops the propagation next phase. Cell doesn't enter next phase. Cell doesn't enter the mitosis. So before mitosis, the quality of DNA is assessed 3 times. Okay, and then inside, unreplicated DNA checkpoint is another checkpoint in the G2 period. So if some regions of DNA is not reduplicated, then cell do not enter mitosis as well. In the mitosis, there are 2 checkpoints: the spindle assembly and chromosome segregation. So we'll discuss them in the next slide. Also, you have to know 2 terms associated with those checkpoints. This is the mitotic catastrophe. This is the impossibility to arrest the cell cycle, and in this way, the aberrant chromosomal number might occur in daughter cells. So such cells, they should enter apoptosis and should be self-destroyed. And the contact inhibition Cell division, it implies detachment of the cell from its surrounding environment. So when cell is detached, it undergoes mitosis. If cell is attached, it doesn't divide. So under normal circumstances, cells, if it is in contact with neighboring, it doesn't divide. Okay, we'll discuss it later. It is important in metastatic cancer. Okay, mitosis. Mitosis itself, you know a lot from biology from school, so let's not stop on details. Of course, in the prophase, chromosome condensation takes place. The protein condensing participates, making chromosome very condensed. Nuclear envelope, nucleolus, they disappear, and kinetochore appears in the region of centromere. Here kinetochore is shown in the diagram. Kinetochore is the protein complex. Then metaphase. Metaphase is a very critical phase because spindle fibers are formed here, and you should know 3 types of spindle fibers. Astral fibers. Astra means a star, so in star-shaped manner those fibers arise from both cell centers. You remember cell centers were reduplicated in the S period. Yeah? So astral microtubules, they arise from cell centers with their plus ends oriented to the cell periphery, and they help to proper put those cell centers. Then polar microtubules, they extend from one pole to another, the contact one with another. Between them there are dynein molecules, and they help to push those cell centers far from each other. So in this way, polar fibers, they help to put cell centers apart to opposite sides of the maternal cell. And also there are kinetochore microtubules. They arise from kinetochore, as kinetochore could be the microtubule organization center. And kinetochore is used to move along those microtubules. But first, what we have to say that remember there are 2 checkpoints in the mitosis. The first checks whether all chromosomes are fixed to the spindle. It is very important because if any chromosome is left unfixed, or if chromosome is fixed with kinetochore fibers to the same pole, such division would result in the aberrant chromosomal number. So as a result, an aneuploidy will be formed, and it is extremely severe pathology, and daughter cells would not be normal. So to prevent that, a special enzyme is used. It is called Aurora B kinase. Along with the metaphase, Aurora B kinase checks every chromosome, and if one chromatid is unfixed, if 2 chromatids are fixed to the same pole, or in case of any another abnormalities, Aurora B kinase, it cut kinetochore fibers and stimulate reappearance of the fibers until every chromosome is properly fixed with one chromatid turned to one pole, another chromatid to another one, because daughter cells should get equal number of chromatids, and 2 sister chromatids, they should always be fixed to opposite pole, and that's what is checked in the checkpoint in the metaphase, and it is assessed by Aurora B kinase and a special protein securin, like security. Yeah, it also checks whether there is a tension in both kinetochore fibers. Okay. So if everything's okay, all chromosomes are fixed, if every sister chromatid is faced to the opposite pole, then the anaphase is allowed, and a special protein, it is called separase. It separates 2 sister chromatids. The cohesin is broken down, and in anaphase, 2 chromatids, they start to move to opposite pole. Movement of chromatids needs ATP energy, and it is due to the movement along the microtubule. At the same time, microtubule is shortening, so tubulin molecules are detached from this region. So microtubule becomes shorter and shorter, while the chromatid is walking along it. Something like chromatid is walking along the bridge, which is broken just behind. Yeah, and eventually all chromatids get separated, and in telophase we can see reappearance of the nuclear envelope, nucleolus, and the cytoplasm is divided, which is called cytokinesis. Cytokinesis is realized because between the two daughter cells a ring of actin and myosin is formed, and contraction of this ring helps in detachment of two cytoplasms. Okay, and here besides the USMLE example is given, and you can't answer, of course, the test, because here the example of Hodgkin lymphoma should be recognized by this histological specimen and by the description of clinical symptoms. But what you have to know is that what treatment should be initiated, and you should understand why vinblastine is prescribed, because vinblastine, like colchicine, it breaks down the mitotic spindle formation, and in this way tumor cells will be will stop their proliferation. So what you should know is that vinblastine, as other alkaloids like colchicine, vincristine, they block mitotic spindle assembly and they block mitosis. In this way they stop proliferation of cancer cells, besides not only cancer cells but rapidly dividing cells as well, of course. Yeah, and here to summarize, this video is shown. The full version could be found here, and the link will be in the description below. So here kinetic core is shown with kinetic core fibers, and you can see that microtubules are elongated and tubulin molecules are added here. Yeah, and in this way every chromosome is put exactly in the center, in the metaphase plate in the center of the cell. And once every chromosome is put properly exactly in the middle in the metaphase plate, and if both chromatids are fixed to opposite poles, you see this green color is the evidence that everything's okay and chromosome is properly put. Then only after that this chromosome gets the signal, and only after that separase would break down cohesin molecule, and only after that two sister chromatids are breaking down, and now they are moved to opposite pole. Okay, so here is another video, the real image of the metaphase plate, anaphase, and the telophase, yeah, the cytokinesis. Here once again, okay, the full video is there. Let's move on. And now what about the cell death? Types of nuclear destruction. First, before we are talking about the cell death, you should be aware about those three terms. What do they mean? Karyolysis. This is the lysis of the nucleus, when nucleus is damaged and its membrane is disintegrated. Then karyopyknosis is when nucleus become condensed and very dark. Karyorrhexis is the type of nucleus destruction, usually occurs after pyknosis, when a nucleus is broken down into small bodies, condensed bodies. So now let's discuss and compare two types of cell death. Actually, there are many more types of cell death, but that's what you have to know. And it is better to compare them one with another to understand the difference and to understand better each of them. So necrosis is something like a violent death by accident, and apoptosis, this is the programmed cell death, like cell suicide, when cell disappears without damaging of its neighboring cells. So what's the difference? First of all, in the death of nucleus, in case of necrosis, the nuclear envelope get damaged, and as a result the nuclear content is spilled in the cytoplasm. In case of apoptosis, Cell is broken down via the with the help of karyotyping and karyorexis. So nucleus become condensed and is broken down into small particles. So its content is not spilled, and if there are any virus, this virus would not infect others. Then DNA, in case of necrosis, is cut into random fragments because it is cut with lysosomal enzymes. So if there are any viral particles or viral information integrated, this viral information could infect others. In case of apoptosis, DNA is fragmented into nucleosomal pieces. So like a paper put through the shredder. In the same way, the DNA is cut into nucleosomal fragments. So if it contains any improper information or mutations or viral information, then virus will be destroyed together with the DNA into small pieces. Then we can see in case of necrosis that cell swelling occurs and plasma membrane becomes disintegrated. So the content of the cell is spilled out and it infects and affects others. While in case of apoptosis, cell is broken down into membrane-bound vesicles. In this way, if there are any poison inside, any toxin, any virus, anything dangerous, then everything's packed into vesicles, and those vesicles are safe for the surrounding environment. Then genetically, the ATP demand. In case of necrosis, it doesn't require ATP because cell is just simply destroyed violently. In case of apoptosis, cell needs ATP to realize this program, to destroy itself safely. It needs ATP. And the last difference is that in case of necrosis, inflammation is launched as the cell content together with lysosomal enzymes, together with different maybe toxins, get released and affects others. While in case of apoptosis, those membrane-bound vesicles, they're called apoptotic bodies. They expose on their surface the phosphatidylserine. This molecule is usually located in the inner phospholipid bilayer, but in case of apoptosis, it appears in the superficial layer of phospholipids. And if phosphatidylserine appears in the outer leaflet, if you want to know more about this molecule, watch the previous lecture. We have already discussed it, So the appearance of phosphatidylserine in the outer leaflet of the plasma membrane, this is something like eat me signal, and it stimulates the macrophages and surrounding cells to engulf those apoptotic bodies. What is interesting in case of apoptosis, if cell is destroyed just only because it is no longer needed, its organelles could be used by others. For example, if there are mitochondria and those mitochondria are not damaged, such mitochondria could be engulfed by neighboring cells and used by them. So apoptosis is very reasonable cell death. And here is another USMLE example, and the question is what process is most likely occurring in the region indicated by arrows, so independent on explanation on this information. You have to understand that here karyotyping and karyorexis takes place, so probably this is the apoptosis, but as you can see, another processes are given here, so you should distinguish them one from another. Okay, and at the end of the lecture we should discuss different pathways of apoptosis, as I think it's very important nowadays because regulation of apoptosis is especially important in treatment of tumors. First, external pathway, when cell is stimulated to undergo apoptosis with the help of another cell. This pathway includes Fas receptor or CD95 molecule, and this receptor, if it is exposed on the cell, if it interacts with its ligand exposed on another cell, for example leukocyte, then if this interaction occurs, then in the cytoplasmic part of the first cell, death domain associated with the disc, with the formation of the disc complex, it activates caspase 8. Caspase 8 activates caspase 3, and this enzyme directly stimulates the apoptosis by breaking down the cytoskeleton, breaking down the nuclear envelope, DNA, and so on. Besides, remember that caspase, these are enzymes characteristic for apoptosis. Then mitochondrial apoptosis. This is the internal pathway. If this pathway is stimulated through the receptor by another cell, then mitochondrial apoptosis. This is when cell itself stimulates apoptosis. How it occurs? There are two protein, Box and Bach, and in case of mitochondria damage, or in case of many problems in the cell, if those two proteins are activated, they form pores in the outer mitochondrial membrane, and through those pores, cytochrome C come out of the mitochondria to the cytoplasm. This is the most critical event. If cytochrome C is released from the mitochondria, then this cytochrome stimulates the formation of the apoptosome. Apoptosome is the complex when cytochrome C activates caspase 9. After that, caspase 9 activates caspase 3. And the third caspase, as I have just said, it stimulates the breakdown of cytoskeleton, nuclear envelope, and DNA into fragments. As a result, this cell enters the apoptosis and realizes the apoptosis here. So you have to remember Box and Bach as pro-apoptotic enzymes, molecules associated with the mitochondria, while BCL2, BCL-XL, these are the two molecules with anti-apoptotic activity, and they prevent this pathway. So as they are detected in some tumor cells in diagnostic purposes and prognostic purposes, you should know their function. They are widely assessed. So Box and Bach, they are pro-apoptotic, while BCL2, BCL-XL, they anti-apoptotic. Let's move on. And those also, apoptosis could be stimulated and triggered by DNA damage. And in these terms, you should know and be aware about the protein, tumor suppressor protein P53. It's very important, and all oncologists and pathologists know what this protein is. This protein assesses the quality of DNA. Let's return to our checkpoint periods of the cell cycle. And you remember that here in the G1 period, the DNA damage checkpoint takes place. Exactly P53 assists in this process. So before entering the S period of the cell cycle, cell assess the integrity of DNA, and if DNA damage is found, then P53 promote cell cycle arrest. It prevents the propagation of the cell cycle. Then it stimulates DNA repair. And in case this repair is ineffective, or there are too many mutations are there in the genome, then P53 stimulates apoptosis. So P53 is very important in prevention of tumors. So that's why it is called tumor suppressor protein. It is not surprising that in 50% of human cancers, P53 is mutated and it is abnormal. Then also let's remember about telomeres. So telomeres, they become shorter and shorter with each next division, and eventually when they exhausted, then cell can't divide anymore, because with next division cell would lose not telomeric region but now codon regions, and it is considered by the cell as the DNA damage, and also p53 stimulates apoptosis in such cells. So telomere mediated cell death is also mediated through the p53 protein. Why p53 is important in diagnostic purposes? Because if it is mutated, then tumor cells are resistant to radiotherapy. You know that sometimes tumors are irradiated. Not only chemotherapy is used to treat tumors, but also radiotherapy. So if p53 is mutated, there is no reason, or there is a low effectiveness of this therapy. So mutations of p53 are assessed before treatment. Yeah. Then, this is the last slide. So, except apoptosis and necrosis, there are many more other types of cell deaths, and it is impossible to describe them all. If you want, you can find lots of interest in the literature, but let's mention the most popular. Anoikis, this is the form of cell death when cell is detached from others and from the extracellular matrix. As I told you, under normal circumstances, cells they should be attached. If they lose the connection with others, with the environment, then such cells are self-destroyed. And in cancer cells it doesn't work, because cancers they send metastasis. Under normal circumstances, metastasis should be self-destroyed because they are not attached to the environment, but they survive. So stimulation of normal apoptosis in cancer cells, this is the purpose of many modern medicine approaches. Then mitotic catastrophe is the type of cell death when cell enter the apoptosis. In case of checkpoints, control checkpoints, the evidence that there are some problems and cell can't propagate through the cell cycle. In this way, cells stop, and if it can't solve the issue, then it is self-destroyed. Then pyroptosis. This is the form of cell death during the inflammation, and it is triggered by different microorganisms. Pyroptosis, it uses caspase-1 as the platform to activate another factors, and is very important, and in pathology department you'll return to that, I believe. Yeah, and entosis is very interesting. It is compared with the cell cannibalism, when one cell eat and engulf another. What is interesting is that this is not the phagocytosis, because this is not a phagocyte. These are not the professional phagocytic cell. Instead, this is one epithelial and another epithelial cell, and that's what's characteristic for cancer as well. So if under the nutrient deprivation, one epithelial cell could eat another one. So okay, this is the last slide of our lecture, and thank you for the attention. If it is interesting, please subscribe our channels, and if you have any questions, then put in the comment below. Thank you for the attention, and goodbye.